PCR amplification is a process by which a scientist can take one segment of DNA and multiply it into billions of copies of that segment of DNA. PCR amplification is done in a centrifuge which has a set of chromosomal DNA, free nucleotides, primers which are targeted for the target segment of DNA that the scientist wants, and something called a Taq polymerase. A Taq polymerase is an enzyme that creates DNA, or attaches DNA, and it is found in hot springs, and we use Taq polymerase because it doesn't denature at high temperatures. Okay, so PCR amplification can take up to a few hours and has to go through 25 to 30 cycles. So the cycles go like this. The first step in a cycle is that the centrifuge tube is heated up to 95 degrees Celsius so that the hydrogen bonds on the DNA denature, and then the second step is that the tube is cooled down to 60 degrees Celsius so that the primers can anneal to their target complementary strand of DNA, and then the sample is increased to 72 degrees Celsius, which is a optimal temperature for the Taq polymerase. The Taq polymerase then begins adding DNA to the segment of DNA. It doesn't just stop on the target DNA though, it adds a bunch of flanking DNA. And then this cycle is repeated 25 to 30 times, so that and with each subsequent cycle there is less flanking DNA and more of the target DNA. After all this is finished, all that's left, or the main thing that's left, is the target DNA, which scientists can use to sequence and do other stuff.